Picture this. You're in the final stretch of a marathon. Legs are burning, lungs are screaming, but you push through, crossing the finish line in a burst of energy. Ever wonder what fuels such incredible feats? The answer is hidden deep inside your cells. We're talking about energy, the invisible force that makes life possible. Your body can't use energy directly. That's where ATP comes in. It's the energy currency of your cells. So buckle up, science fans, as we dive deep into the fascinating world of ATP, the secret fuel that keeps you alive and kicking. ATP stands for adenosine triphosphate. Think of ATP as tiny rechargeable batteries floating around inside your cells. Each stride, each heartbeat, each gulp of air, all powered by ATP. ATP stores energy in the bonds between its phosphate groups. When these bonds are broken, energy is released for cells to use. Your body constantly recycles ATP, adding phosphate groups back on to store more energy, ready for action. Imagine your cells as bustling cities filled with factories, transportation systems, and communication networks. This is where ATP comes into play. Think of ATP as the currency of these cellular cities. When you eat, your body breaks down the food into smaller molecules like glucose, which contain stored energy. This energy is then used to create ATP, ensuring a constant and readily available energy supply for all of life's activities. So what exactly is ATP made of? Despite its vital role, ATP is a relatively simple molecule. It's composed of three main parts: adenine, a nitrogenous base, ribose, a sugar, and three phosphate groups. The real magic lies in the three phosphate groups. These phosphates are negatively charged, storing a significant amount of energy in their bonds. When a cell needs energy, it breaks the bond between the second and third phosphate groups, releasing the stored energy. The energy released from breaking the phosphate bond in ATP is energy in a form that cells can directly use. This energy is used to drive a wide variety of cellular processes, from muscle contractions to nerve impulses. When you contract your muscles to lift a weight, specialized proteins in your muscle cells change shape, pulling the muscle fibers closer together. Similarly, when you think or feel, nerve cells communicate with each other through electrical and chemical signals. Without ATP, these processes would grind to a halt, like a city plunged into darkness without electricity. Here's the amazing part. ATP isn't a one-time use energy source. It's constantly being recycled in your cells, like a rechargeable battery. Once ATP breaks down into ADP and Pi, releasing energy, your cells can use the energy from food to reattach the phosphate group to ADP, creating a fresh molecule of ATP. This continuous cycle of ATP breakdown and regeneration is called the ATP cycle. It's a fundamental process that occurs billions of times every second in every cell of your body. The beauty of ATP lies in its versatility. It's involved in a breathtaking symphony of actions that keep you alive and thriving. From the rhythmic beating of your heart to the complex computations in your brain, ATP is the driving force behind it all. Muscle contraction. Each movement you make relies on the coordinated contraction and relaxation of your muscles. Nerve impulse transmission. Your nervous system relies on electrical and chemical signals to transmit information. Active transport cells often need to move molecules against their concentration gradients, and ATP provides the fuel for this. So far we've explored the concept of ATP as the energy currency of our cells, but where does this vital currency come from? The answer lies in a fundamental metabolic pathway called glycolysis. Glycolysis is the breakdown of glucose, a simple sugar that serves as our primary energy source. This process occurs in the cytoplasm of every cell, the bustling hub where countless biochemical reactions take place. Glycolysis sets the stage for subsequent pathways that yield a far greater energy harvest. Now let's zoom in on the intricate dance of molecules that defines glycolysis. It's a multi-step process, involving a series of enzymatic reactions that systematically dismantle glucose. The glycolysis pathway can be divided into two main phases: the energy investment phase and the energy payoff phase. During the energy investment phase, the cell invests a small amount of ATP, priming the glucose molecule for energy extraction. The second phase, the energy payoff phase, is where the cell reaps the rewards of its investment, generating a net gain of ATP. As we delve into the energy investment phase, imagine glucose entering the cellular arena, ready for its transformation. The first few steps involve adding phosphate groups to the glucose molecule, a process called phosphorylation. These phosphate additions trap glucose inside the cell and destabilize the glucose molecule, making it more reactive. Phosphorylation creates weak points in the glucose molecule, making it easier to break apart in later steps. This initial investment primes the glucose molecule, preparing it for the energy extraction to come. With glucose primed and ready, the glycolysis pathway shifts gears into the energy payoff phase. Here, the modified glucose molecule is cleaved into two smaller molecules called pyruvate, releasing energy. This released energy is captured by electron carriers, molecules that ferry high-energy electrons to other parts of the cell. The energy payoff phase also yields a small but significant direct payout of ATP. By the end of glycolysis, the initial glucose molecule has been fully dismantled, yielding a net gain of 2 ATP molecules. As we delve deeper into the intricacies of cellular respiration, imagine glycolysis as a bustling marketplace. Pyruvate, like a traveler arriving at a border crossing, must undergo a crucial transformation before it can enter the next stage of energy extraction, the Krebs Cycle. The Krebs Cycle is a grand, rotating wheel where molecules undergo a series of chemical transformations. Before pyruvate can join this ride, it must be converted into Acetyl-CoA, a molecule that acts as the key to unlock the gates of the Krebs Cycle. This conversion process involves removing a carbon atom from pyruvate, releasing carbon dioxide and generating a molecule of NADH. With Acetyl-CoA in hand, the adventure continues as we step onto the platform of the Krebs Cycle. The cycle begins as Acetyl-CoA merges with a 4-carbon molecule called oxaloacetate, forming a 6-carbon molecule called citrate. From here, the cycle embarks on a series of rearrangements, oxidations and reductions. As the cycle progresses, carbon atoms are sequentially removed from the intermediates, forming carbon dioxide. These carbon atoms, once part of the glucose molecule, are now released into the cellular environment, their energy potential harvested and transferred to electron carriers. While the Krebs cycle might seem like a complex and intricate dance of molecules, its primary goal is surprisingly straightforward: to extract as much energy as possible from the remnants of glucose, those 2 carbon units carried by Acetyl-CoA. And how does it achieve this feat? By systematically oxidizing the intermediates of the cycle, stripping away electrons and transferring them to those ever-reliable electron carriers, NADH and FADH2. Think of NADH and FAD2 as cellular buses picking up passengers' electrons at various stops along the Krebs cycle route. Each time a molecule in the cycle is oxidized, it loses electrons, and these electrons are scooped up by NAD+ and FAD, converting them into their energy-rich forms, NADH and FAD2. These loaded buses then depart the Krebs cycle, heading towards their final destination, the Electron Transport Chain, the grand finale of cellular respiration where the real ATP bonanza awaits. But the Krebs cycle isn't just about shuttling electrons around, it also generates a small amount of ATP directly through substrate-level phosphorylation. That process we encountered in glycolysis, where a phosphate group is transferred from an energy-rich intermediate to ADP, forming ATP. It's like finding a few extra coins tucked away in your pocket, a small but welcome bonus on top of the main energy haul. While ATP production might be the star of the show, the Krebs cycle is more than just an energy factory. It's a metabolic hub, a central player in a network of interconnected pathways that keep the cellular machinery humming along. Think of it as a bustling city center with traffic flowing in and out connecting various parts of the metropolis. One of the key roles of the Krebs cycle is its involvement in the biosynthesis of essential molecules. Intermediates of the cycle can be siphoned off, like diverting traffic from a busy intersection, to serve as building blocks for amino acids, the precursors to proteins, as well as nucleotides, the components of DNA and RNA. It's like a resourceful city using materials from its central hub to construct new buildings and infrastructure. Moreover, the Krebs cycle plays a crucial role in regulating cellular metabolism, acting like a sensitive sensor that monitors energy levels and adjusts metabolic flux accordingly. When energy levels are high, the cycle slows down, like easing traffic congestion during off-peak hours. Conversely, when energy demands increase, the cycle revs up, like opening up additional lanes on a highway to accommodate rush-hour traffic. This intricate balance, this constant interplay between energy production, biosynthesis, and metabolic regulation, highlights the remarkable elegance and efficiency of the Krebs cycle, a metabolic marvel that underpins the very essence of life itself. We've journeyed through glycolysis and the Krebs cycle, dismantling glucose and capturing its energy. Now the electron transport chain awaits within the mitochondria, our cells' powerhouses. Imagine a stadium buzzing with anticipation, as glycolysis and the Krebs cycle warm up the crowd. NADH and FAD2, loaded with high-energy electrons, are ready to release their energy. This energy powers ATP synthesis, the final stage of cellular respiration. Imagine the electron transport chain as a relay race. Protein complexes in the inner mitochondrial membrane pass electrons like batons, releasing energy with each transfer. NADH and FAD2 deliver high-energy electrons to the chain, starting the race. NADH dehydrogenase accepts electrons, oxidizing NADH to NAD+. Electrons pass down the chain, releasing energy. This energy pumps protons across the membrane. Proton pumps act like turnstiles, creating a gradient. Electrons move through complexes, driving energy conversion. With each electron transfer, the inner membrane space becomes increasingly packed with protons, like a crowded stadium filling up with excited fans. This accumulation of positive charge creates an electrochemical gradient across the inner mitochondrial membrane, a difference in both charge and proton concentration, like a dam holding back a torrent of water. This gradient, however, is not merely a byproduct of the electron transport chain, it's the very force that drives ATP synthesis. The potential energy stored within this gradient, like the water held back by a dam, is harnessed by a remarkable enzyme called ATP synthase, the molecular turbine that powers the final step in cellular respiration. Imagine a water wheel positioned beneath a waterfall, the force of the rushing water causing it to spin and generate mechanical energy. Similarly, ATP synthase sits embedded within the inner mitochondrial membrane, its structure spanning the membrane like a bridge connecting the inner membrane space to the mitochondrial matrix. The flow of protons down their concentration gradient through ATP synthase provides the mechanical energy to drive the synthesis of ATP. This elegant mechanism, this coupling of electron transport to proton pumping to ATP synthesis, is a testament to the remarkable efficiency and ingenuity of cellular evolution. It's a prime example of how cells have learned to harness the power of gradients, those differences in concentration or charge, to drive essential life processes. ATP synthase, the star of the ATP synthesis show, is a remarkable molecular machine, a testament to the elegance and efficiency of biological nanoengineering. Imagine a tiny turbine, its rotor spinning gracefully as protons flow through it, their kinetic energy converted into the mechanical energy that drives the synthesis of ATP. This molecular marvel consists of two main components, F0, the membrane-bound unit that forms the proton channel, and F1, the catalytic unit that protrudes into the mitochondrial matrix and houses the active sites for ATP synthesis. Think of F0 as the turbine's intake pipe, channeling the flow of protons, while F1 acts as the generator, converting the mechanical energy of proton flow into the chemical energy of ATP. As protons flow through F0, their movement causes the rotor, a central stalk connected to F1, to spin like a carousel. This rotation induces conformational changes in F1, altering the shape of its catalytic subunits and bringing ADP and inorganic phosphate, PI, into close proximity, facilitating their union to form ATP. It's like a carefully choreographed dance, the spinning rotor dictating the steps of ATP synthesis. This elegant rotary mechanism allows for the continuous production of ATP as long as a proton gradient is maintained across the inner mitochondrial membrane. It's a prime example of how evolution has favored efficient and elegant solutions, harnessing the power of gradients to drive essential life processes. Section 5. The final tally. Maximum ATP production. As the electron transport chain reaches its crescendo and ATP synthase churns out a steady stream of ATP, let's take a moment to appreciate the impressive efficiency of this cellular power plant. The complete oxidation of one molecule of glucose, through the combined efforts of glycolysis, the Krebs cycle, and the electron transport chain, can yield a remarkable 36 to 38 molecules of ATP, a testament to the intricate and elegant dance of energy conversions within our cells. This remarkable yield highlights the importance of the electron transport chain as the ultimate ATP-generating powerhouse of cellular respiration. While glycolysis and the Krebs cycle contribute a small but crucial amount of ATP, it's the electron transport chain, with its ingenious coupling of electron flow to proton pumping to ATP synthesis, that maximizes the energy harvest from glucose. This efficient energy production underpins our very existence, powering everything from muscle contractions to nerve impulses to the synthesis of new molecules. It's a reminder of the intricate and elegant processes happening within us every second, a symphony of molecular events orchestrated to sustain life. Section 1. ATP, powering every facet of life. ATP fuels our every move, thought, and breath. It's the energy currency of our cells. Why is ATP crucial? Imagine your body as a city. ATP is the electricity that powers it. Without ATP, everything stops. Every heartbeat, breath, and thought relies on ATP. Without it, the world fades to black. Section 2. The ATP edge, from peak performance to aging gracefully. ATP is vital for peak performance and aging gracefully. Athletes need it for muscle power, students for brain function, and everyone for longevity. Maintaining ATP levels through diet and exercise supports health, vitality, and cognitive function throughout life. Embracing the energy of life. ATP powers our every move, thought, and breath. It's the energy behind our heartbeat, the fuel for our muscles, and the spark for our thoughts. Embrace the power within and live each day to the fullest. Like and subscribe for more amazing science adventures.